So the next speaker is Dr. Tong.
Hi, I'm Tong. I'm based in Singapore. I'm a Catalyst Awardee from 2020, funded by Singapore's National Research Foundation and Ministry of Health. So our project is to develop a diagnostic device for Parkinson's disease. So Parkinson's disease is a movement disorder characterized by tremors, rigidity, among other symptoms. It has a very long latent period, and the diagnosis is current method of diagnosis is based on clinical observation, which can be subjective, and there are additional challenges that there are other diseases or disorders with similar symptoms. So one way the clinician would have to confirm diagnosis would be to put the patient on a course of drugs that boost the dopaminergic system. So the motor symptom comes from dopamine secreting neurons dying in a brain region called substantia nigra. So the drugs would work to boost the dopamine secretion in that system in the brain. So if a patient responds positively to the drug, it's only then the diagnosis is confirmed. So given this kind of challenge, there is definitely a strong need for an objective diagnosis system, which we currently do not have. So I am trained as a geneticist, actually, and I work with model organisms like fruit flies and killifish. So this the development of a diagnostic device is not naturally my field, but I came from when I was doing my postdoc, I was in an olfactory lab that studies the olfaction system. So it became, so it prepared me for coming up with for the ideas when I saw my colleague presenting something that turns out to be relevant to what we do. So I have a colleague in Temasek Life Sciences Laboratory that founded a startup a long time ago, and he founded a startup in the US, but he moved to Singapore. So that startup developed an array of 70 gel dots, and each of the gel dots, if you can see in my background, I'm actually trying to show the gel dots on my on my left hand side, right? So so there is actually basically a glass slide with 70 gel dots, and each of the dots have different physical and chemical property, so that when you put urine with that slide, different types of metabolites will get differentially retained among the 70 dots. So that's the technology that the startup developed. But in its original state, it is not able to help us differentiate between urine with Parkinson's disease and normal urine. So what we have been working on is to gather from the literature what are the findings from the metabolomic studies that show certain compounds that are enriched in Parkinson's disease. We will take those compounds and spike it into normal urine and test that on the array. And on top of that is after we let the array capture the metabolites, we need to develop a color reaction, which currently the array is not adequate for. So we have a chemist who joined my lab since 2 years ago. He's been testing various chemical reactions to develop a difference between Parkinson's disease simulated urine and normal urine. So at this point, we are at a point where we are starting to see, starting to have several candidate reactions that can show a difference. And we are about to test combinations of those reactions to see if we can further accentuate the differences. And only when we are able to demonstrate a convincing difference using this method that we are developing, then we will move into clinical trial. And I'll be happy to hear feedback from you.
All right, great. Dr. Koh, any feedback from other attendees?
I have a question.
Oh, yes, Gonzalo. Yes, please.
How are you choosing the metabolites that you're using for the device, and how many do you think could be a good number for identify the disease?
So currently we are using 70 metabolites. Ideally, more metabolites will be better. What's limiting us right now is the number of metabolites that has already been identified using metabolomic studies, and also the commercial availability and the solubility of the metabolites. I should add that after we develop the color difference, we are also developing a computational method to tell the difference. Currently, what we are using is just a PCA analysis, and I am collaborating with a colleague who's doing the analysis, and his take is if we can show the difference using a hierarchical method rather than machine learning, it means that the difference is substantial, and that is that would give us stronger evidence to go forward from here. I think I see a question in the chat box.
Yes, so there's one from Zhao Zhidong. I think he wants to ask about whether other neurodegenerative diseases are also included to compare with Parkinson's disease patients.
I think that is a good idea. Currently, we are definitely not in clinical trial stage, but I think, yes, there is concern that we could be seeing aging markers instead of Parkinson's markers, right? So it would be good if we are able to spike urine that are enriched in elderly donor and compare that with the baseline urine we have, and see whether that is different from the simulated Parkinson's urine. And similarly, we should probably look for compounds that are enriched in Alzheimer's disease urine and spike that into our baseline urine and see whether, and do a comparison. Yes.
Right. So, Dr. Koh, do you expect these associations to be differ across ethnicity, or you think that they're probably behaving in the same way across different groups?
Potentially, yes, and potentially it could be different across people having different diet too. So in general, for urine diagnostics, oftentimes there is a strict protocol as to at which day, time of the day, and whether you're fasted or not when you collect the urine. Currently, the metabolomic studies that we have been looking at are predominantly derived from Chinese population, which happen to be also coinciding with the major demographic in Singapore.
Right. All right. All right. Any other questions from the floor before we move on to the next speaker? All right. If not, then let me move on to the next speaker.